So let's take a look then at some fundamental what we're trying to, first of all, what we're trying to achieve with these forced degradation studies. Now, ideally, what we're trying to do when we set up a forced degradation study is to predict the principal degradation products of our drug substance that we will find in our formulated drug product at end of shelf life. Okay, so taking the formulation out of the equation and thinking only about mechanisms such as hydrolysis and oxidation, what are we likely to see at end of shelf life in our drug product? So, likely degradation modes, and we also, of course, use stress studies to validate the stability indicating nature of methods. So, potency assays should be capable of detecting the disappearance of the drug substance, related substances methods capable of detecting and quantifying the growth of degradation products. So US FDA published some guidance some while ago on what it counted as a stability indicating method. So a validated quantitative method can detect changes with time in the pertinent properties of drug substance and drug product. So it talks about disappearance of the active and appearance of degradation products. It also says something remarkably stupid. It says assay analytical procedures for stability studies should be stability indicating unless scientifically justified. So I would love to see the scientific justification for using a non-stability indicating assay method for a stability study. That would be pushing credibility, wouldn't it? So we're not going to do that. So let's imagine that we are developing a suite of test methods for an entirely new chemical entity. So we don't know where those degradation products are likely to crop out. So first priority then is to develop a test method that's capable of detecting those degradation products. Until we've done that, there's really not very much we can do in terms of drawing conclusions regarding the stability of formulations and so on. So until we have confidence that we are capable of detecting the degradation products that form from a particular drug substance or from a particular formulation, we're not very well placed to conclude anything regarding the stability of that drug substance or drug product. So as we've seen with the previous example of that aromatic group with the nitro functionality and the ester, degradation chemistry can be extremely complex, and a relatively simple structure can give you a variety of different degradation products. So all the various modes of degradation, so temperature, oxygen, so on and so forth, can lead to different degradation mechanisms and different degradation products. And this is complicated further by the conditions that we choose to use for the stress study. Before we go on then to which conditions we should be using for stress studies, let's think about when we might like to perform those stress studies. So, as we're saying, we probably want to be performing these studies in the earlier stages of drug development, certainly for a new chemical entity. And if we're developing a generic drug, and we don't have a good knowledge of that drug substance's degradation chemistry, we'd want to do a similar thing there. And for me, the key thing on this slide is that final bullet point. What we're trying to do at this stage in the context of stability studies is to reduce the risk that our test methods are not detecting an important degradation product. And by the way, just because you're using a compendial method does not necessarily get you out of the woods on this one, because we don't know what particular formulation that compendial method was developed against. If we're talking about a compendial method for a drug product, say a USP method for a tablet formulation, we cannot assume that that particular compendial method will be suitable for our product. In other words, there may be degradation pathways that exist in our formulation that did not exist in the reference listed drug. So a pragmatic approach then, or if you like, a sort of risk-based approach to running stress studies would be to conduct an informal stress study during the earliest stage in development. And then in later stage, as we move from phase 1 to later clinical stages, we would probably want at that stage to conduct a formal stress study to confirm that our test methods were stability indicating. But we should have enough information at that stage to know that that's a low risk.